All right. Well, in this second part of sort of a review of basic genetic terminology as far as modes of inheritance, we're going to move slightly up the scale and remind you about some that hopefully you will remember, but if you don't, they're ones that are, well, I think we'll find are hopefully interesting. All right, so let's take our second look. So we already dealt with the dominant recessive, codominant, and incompletely dominant. And I want to mention lethals. Lethals turned out to be really important in human systems. And we will be talking a lot about things that are lethals in various ways, okay? There's two big categories. These are known as dominant lethals and recessive lethals, okay? Huntington's disease is one example of a dominant lethal, which means that if you get one copy, you have expression of it. Progeria, which is this early aging, these individuals that have a lot of different outcomes. It tends to be a dominant lethal because even one copy of it will lead to the pretty much expression of the phenotypes, ultimately to death. Recessive lethals, icterosis, congenital or full, is this individual here has this skin that's ichthyo is scaly skins, so skins where you do not have the right formation of a skin pattern. This tends to be a recessive lethal. In other words, you don't see the full expression of it in individuals. So let's look, just kind of remind you of these. They're difficult ones for students to sometimes grasp, so just kind of remind you about it. So a lethal gene is any allele that results in death. In ecology parlance, it really just prevents reproduction, right? But genetically, it's when it's probably going to kill you. But the idea is that genes have to move to the next generation. So if a gene stops that, it's might as well be a lethal gene. Okay. And here's what's really a problem for people is that the true meaning of dominant recessive. Recessive means that you get two copies. It doesn't matter, and I just stress this over and over again in general genetics, but it doesn't matter what you call them, right? Big Q's, big T's, little T's. If two copies of them show up, okay, that's recessive. It takes those two copies to be present, however you label them, 1 and 1 and 3 and 3. That's recessive. A dominant is one that only takes one copy. Doesn't matter what we call them, okay? And that's important because we start talking about these lethals. We'll talk about pleiotropy and things where it's one side of the equation, it's this, the other side it's a recessive lethal or whatever, okay, or a dominant lethal. So what is a recessive lethal? It's a lethal allele that's expressed only in the homozygous form, okay? So, it doesn't matter whether how you express the form or how you write it, I should say. That's the dominant. That's the only time it's going to get you is when you get a copy from both mom and both dad. You get maternal and paternal copies give you that form. A dominant lethal is a much more difficult one to deal with because it doesn't matter whether you're homozygous or heterozygous, right? If you get one copy of that form, ultimately it's going to lead to death. That's the idea. So it doesn't matter what the other parent gives you. It's just a classic dominant form. It works there, okay? All right, so dominant lethals. Well, what do we know about them? Well, they're fairly rare in some senses. Well, it will turn out in humans there actually are quite a few, but they don't get, if it's gonna kill you, and it takes one copy to kill you, it's not likely that you're going to make it reproductive age, particularly in humans, which we have this delayed adolescence, right? Maybe it's quite a few years before we become reproductively functional, and certainly biologically, and then in society, we may even be longer than that before we tend to be reproductively our life cycle. Something could happen, is going to happen to you in that time, okay? Now, I've drawn them up here like this. I do that to torture the undergraduates because they're like, wait, little a, how can that be? How can little a, that doesn't make any sense. How can that be a dominant lethal when it's little a's? Well, because again, following the instructions of what we said, if you get one copy, you're dead. You get two copies, you're dead. Doesn't matter what the other expression of it is, okay? And the big point of that is there's usually, we don't have very many killer genes, right? There are some senescence genes that tell cells it's time to shut down or tissues it's time to shut down. But we don't, think about an evolutionary sense, why do you evolve a gene that's going to kill you? You don't really evolve those genes. What happens is you've got some normal functioning gene over here, and there's a mutation in it that either shortens it or makes it non-functional or some other way, whether it's point mutation or some larger scale mutation. And then it means that you just don't produce that protein properly, or the protein's misfolded or something else, and it so overwhelms the system that it kills you. Okay, that's really what it is. Okay, um, in that perspective, true dominant lethals usually only last one generation. Right? They're not gonna have them last very long, because that's true. Because if it kills you, um, and you've got it, you're gonna die. Right? It's usually only gonna last one generation. Because even if you're a het, you're going to die from it, right? And you're not likely to pass it on. If you're homozygous, you're definitely going to die from it. And even if you do pass it on, you can only pass on the stuff that kills your kids, right? So that's usually the thought about it. So how do we have these dominant lethals? Well, there are lots of ways, and we will talk some more about them. But in general genetics, we always talk about things like delayed expression. That's a classic form. Huntington's disease, Huntington's chorea, is a classic dominant lethal. If you get the mutated form, Huntington's allele, you're going to die from it. It's going to kill you. Okay. Now, if you're homozygous, which is very rare because it's a very rare allele, very rare disease, at least in the homozygous form, it's lethal. They generally don't even make it to term, or if they are, they're very short-lived individuals once they've had parturition. If you get the het, which is what you see most people being, is that they're fine up until they get into their 30s and 40s and maybe 50s. And then they just start developing this whole suite of related problems because they get this buildup of these external bodies. Basically what they're doing is they're producing too much protein, and the systems that are responsible for breaking those proteins down aren't functioning. So, amyloid beta being one of the proteins. And so particularly in the brain or neural system, the cells go, well, what do we want to do with this protein? Well, they just stick it on the outside, kind of like in your arteries, cholesterol stuck on the outside. Well, they stick them on the outside. Well, if you're a neural cell, and we'll see this for lots of diseases, if you're a neural cell and you're trying to fire between two and there's a bunch of gunk stuck on the outside, it's not going to work very well. That happens in Huntington's. Is basically what ultimately happens. In the heterozygous condition, takes a number of years for that to actually come to full effect, right? But it will eventually kill you. Now, of course, if you have made it into your 30s or certainly in your 40s, if you're going to reproduce, you've likely reproduced to that point. So, you can pass it on. So hets, where we see it getting passed from generation to generation is through these individuals that are heterozygous. Okay? There's a couple other things that go along here. When we talk about whether we're talking about a recessive lethal or a dominant lethal, you have things called sublethality or semi-lethals. Sublethals are things that, it takes very strong conditions for those to kill you. They have to interact with something else. A semi-lethal is kind of the same form. They have different terminologies in the medical world, but they basically mean that you may not die from it, okay, even though you show up with it. And that's really important in things like genetic markers. We have these things, we always talk about in genetics is expressivity, whether the gene is actually going to get expressed. So just because you get this genotype here, okay, is it going to actually express itself or not, right? And you may have expressivity, 100% for some genes. You may variants, and you may have expressivity of 40%. Just depends on how the cellular conditions work out. And you can have exposure, right? There's some genes that are going to be lethal if you're exposed to certain conditions. If you never get exposed to asbestos or something, then you may have a genotype that, even though you have a genotype that if you were exposed, you might respond as a death. Cigarette smoke, whatever chemical external forms that might have. Those are part of those. So we'll talk more about those as we highlight sometimes when those occur. Okay. All right, and then the other two I want to talk about here are things like they'll remind you about are sex-linked. Okay. And sex-linked come in two forms, and particularly with humans, we talk specifically about X-linked. Okay, so that's a form of sex-linked. Remember, in humans, with our chromosomes, we've got the X chromosome is the largest of the two sex chromosomes, and so obviously it's going to have the most genes on it because it's the biggest. So you're going to have the most of the sex-linked variations are going to be in the X chromosome. One of them, the classic one we look at, is color blindness, right? Color blindness is a sex-linked trait, and we see more males that are colorblind because males are hemizygous, and so if you get color blindness from mom, you know, there's the X, that's where it comes from, right? In a male, they're going to be colorblind, you know, if it comes that way. It takes, now you can see colorblind females. My best friend says she's colorblind, and she's, you know, which tells me immediately that her dad was colorblind, that her mom is not colorblind, but she had to have been a carrier, right? So she must have been a heterozygote. And I guarantee that my godson is colorblind, right? Because he got an X from mom. The only X mom could give him was colorblindness. Various forms of hemophilia tend to be X-linked. And then there are things that are, of course, sex-linked on the Y chromosome. We give those a special name. Okay, we call those holandric, and holandric means they're passed from father to son because that's where the Ys are passed. Things, you know, there's not a lot of genes there. There's some important ones, but things like testes determining factor. So the genes for making testes are actually autosomal. They're not in the sex chromosomes, but the genes that control the regulatory function of those, turning those genes on at the right time during development, are actually testicle determining genes are found on the Y chromosome. So you know, and there's lots of stuff. If you look at the X chromosome, it's got about what we think right now from the, you know, our 20,000 and 19 to 20,000 gene functions is there's about 874 or 875 protein genes there. I'm still finding some. There's a lot of RNA genes. Okay, remember that, you know, we talked about the fact that a lot of our genes don't make, all genes make RNA. Only some of that makes coding proteins because it's translated. Others make RNA, various forms, tRNAs and smRNAs and all the other forms. And what's really cool is there's a whole bunch of pseudogenes there. There's more, as many or more broken down genes on the X. It seems like it's become kind of a somewhat dumping area. And that's because over evolutionary time, you know, the X chromosome has been very important in sex determination, and there's things that get, you know, genes that are there that get turned off or moved there. The Y chromosome has very few, as you expect, because only half your population is going to have a Y, so you don't want to put, you know, many genes there. But there's about 73 or so genes that are found there. A lot of those are, again, to do with secondary sex characteristics and other determining factors. Some of them, the X and Y, even though they're different, they do have a region on the bottom of the X and the top of the Y where they can synapse. So they actually have shared genes in a small region that works. So that, you know, part of those genes are actually ones that are cross both. So in that small region, you actually then do have things like codominance, incomplete dominance, and all that, because you got both copies are there. You don't have the holandric effect. There's some RNA genes. These are copies of genes that are found in other places. You know, they're not unique to the Y chromosome. And there's about 400, which is a lot considering it's a tiny little chromosome. You know, it's now there's no recombination, right? Because except for that little piece that fits there on the X, there's nothing to recombine with. So you don't see any recombination on the Y chromosome. So it's kind of an interesting form, right? And two of the others, the last two that I'm going to do for this lecture, and we'll move on to the other ones in the third review, are these two that we call sex influenced and sex limited. Again, these are ones that just, we don't mean to torture the general genetic students, but these do. The sex linked, it's physically on one of the sex determining chromosomes. Sex influenced and sex limited are autosomal. You get two copies. It just depends on how the cell, as I try to anthropomorphize it, is the cell looks over to see whether you're a male or a female, and then determines what you're going to do. Of course, look, it looks at the expression patterns and all that. But basically, sex limited is what it says, right? It's things that are autosomal, but if you're a male, you don't express them. Or if you're a female, you don't express them, or you do express them, okay? Primary and secondary sex characteristics are classics of this. We're all mammals, all humans are mammals, right? Therefore, we have the ability to produce milk, right? We have the genes to lactate, okay? As males, we don't tend to lactate. Okay, there are some exceptions for things, but we don't develop the plumbing to lactate. We don't develop those characteristics. Even though we carry the genes, they're never expressed. They're cut off in development. Okay, testes development, etc. Those are limited. So we have all sorts of things, hair patterns and things. Now, baldness, like me, okay, that's not a sex limited or that's not a dominant recessive trait. I have to explain all the time to people that no, it's not about your grandmother or granddad having bald and you're not. It's sex influence. There are bald females, there are bald males. Okay, there's a balding gene. It's just that the heterozygous condition in males results in being bald. The heterozygous condition in females doesn't tend to do that. Okay, and it has to do with testosterone in the system, presence or absence of testosterone driving system. Now there are balding females. Okay, homozygous bald will be individuals, and they'll tend to lose hair in the back and things. Okay, homozygous males for the balding will be very bald early, quickly. So just remember, sex influence, sex limited. Not a lot of those we're going to talk about, but they're kind of ones you just kind of remember. Okay. All right. Well, that's the second review. Those are the last of those. Again, we're almost all these cases we're talking about, or in fact, all these cases, we're talking about a single locus system where we're dealing with just the variation that occurs at those two loci, or excuse me, at that one locus, at those two genes, and how they relate to each other as far as production into the pathways. The next thing we'll talk about, the last of these that we'll remind you about, are ones where you get more complicated. You have multiple loci interplaying with each other, and we're getting now toward the real world.